And with that, Daniel, the floor is yours.
Hello, everyone. I hope you're all having a great day. Thank you for joining me and everyone else today to listen to our science talks about parasites. Today, I will be talking about the structure and function analyses of trypanosoma protein known as KREL1. We'll get into more about that. Let's see. Okay, so let's start with a case study. So a 67-year-old man, retired engineer, has been an engineer for 40 years, decides to take his skill and volunteer in Ghana to build homes and schools. And each year he goes there and he gets these bites all over, but he thought nothing of it. And then after 4 years, this man started to become more confused, drowsy, started to have an unstable gait. His cognitive deterioration started, becoming more confused. So he went to the hospital and they did an MRI and they saw many lesions of his brain. And what did they diagnose him with? Well, antibodies basically diagnosed him with Trypanosoma brucei, causing human African sleeping sickness. So this man went through 3 years of treatment, long treatment, and physiotherapy. And luckily, this man survived, but not everyone is as lucky as the 67-year-old who, had human African trypanomyosis. And so that's why my work, it focuses on parasitology, but also biochemistry, which is the proteins involved in these parasites, as means of drug targets, as mean of a target for drugs against these parasites. Which parasites do I work on? I work on a group of parasites known as Tripuramacidae protozoans. They cause debilitating diseases around the world, such as human African trypanomyosis, like I've mentioned. But you also have Trypanosoma cruzi and Leishmania species. And collectively, there's 10 million cases around the world. There's drug development, but there's resistance that is increasing. There's side effects, but there is no vaccine. And so there's a lot of research that needs to go into different targets against these parasites. Let's look at the life cycle of these interesting parasites. So this 67-year-old man, when he went to visit Africa, like I said, he had these insect bites. Well, that's essentially how it starts off. A mosquito will bite you. And in the bite, it may transmit this parasite. It may not, but at times it may will if the mosquito has the parasite when it bites you. So when the parasite goes into your bloodstream, it can go in many different areas of your body. If it goes to the brain, it can cause human African trypanomyosis and lead to cognitive deterioration. If it goes to the heart or the GI, it can cause Chagas disease and lead to problems in the heart and the GI tract. It can also go to the liver or just cause blisters all over the body. And we see this in leishmaniasis, okay? Let's take a look at what's going on when the insect actually goes into your skin. After 5 hours, it does something known as binary fission. Essentially, it goes from one parasite to two. It's only 5 hours. After one day, it may look like this. And after 1 month, just by that binary fission, look at the mass amount of parasites and multiplication factor. You can imagine how that would cause heart and liver and GI problems just by having the sheer amount of parasites in your blood system. If we look at these parasites underneath a microscope and we stain them, we end up seeing a dense structure. Well, this structure is known as the kinetoplast. This kinetoplast is actually the DNA of these parasites. So while our DNA is arranged in these chromosomes, these parasites have their DNA arranged in these intercalated mini and maxi circles. And this DNA will encode for RNA, which then needs to get edited to make functional proteins that you see right here. Those functional proteins allow that parasite to divide. So wait, what did I just say? That DNA needs to go to RNA, which needs to be edited in order to make functional proteins. And so my research essentially wants to prevent that editing process. If we don't edit the RNA, we don't have functional protein. We can't get that sheer amount of parasites in our heart, in our GI, in our brain, just like in the 67-year-old man. And this RNA editing process involves an essential, a very important protein known as Kinetoplastid RNA editing ligase 1. And that is the protein, that is the biochemistry of my research that I focus on. And that's essentially how I bring biochemistry and parasitology together. So let's look a little into my research. In 2004, what was published was this is the full length of my protein. And representing activity is a black band, okay? We won't go in details as to what that is. Activity is known as adenylation. It's just binding to ATP. But whatever that is, it's represented by a black band. And so what we see here is when you have the full-length protein, you get activity. You get a nice black band. However, when they remove half of the protein, when they remove the C-terminal domain and they truncate it, there's very little activity. In fact, this is actually enhanced 16-fold. So you can almost say activity is abolished. So what is going on? What is it in that C-terminal domain that is responsible for the loss of activity? That may be an interesting drug target, right? That's my research. So what I did to narrow down that important region was I performed mutations where I kind of cut off piece by piece of the C-terminal domain, slowly but surely truncated the C-terminal domain, just like this. And so the full-length protein is 469 amino acids. I end up making proteins that are shorter and shorter, 462, 54, 46, 40, and eventually what you see, which is just the N-terminal domain, which we know is inactive, like I showed. Once I purified and expressed these proteins, I then tested them in that similar assay. But today I'm going to show you in a graph formation. And what we have compared to the wild type, when looking at activity, we have these three truncations, which are relatively similar to the wild type. However, as soon as I truncate at amino acid 440, activity is abolished. Whoa, what just happened? Because before, as I said, when you take away the whole C-terminus, activity is lost. But now I've kind of truncated slowly but surely and realized that once I truncate after amino acid 440, that's when activity is lost. So what do I want to do next? Well, I want to look into that region. So that's what I did. Because now I'm only looking at really 6 amino acids, right? Because we had function with this mutant, we lost function with this mutant. And if we look at those 6 amino acids, we look at Trypanosoma brucei, the one that caused human African trypanomyosis, you see a lysine, a tryptophan, a lysine, and a glutamic acid. This is actually conserved in Trypanosoma cruzi and very similarly conserved in Leishmania. The reason why it's in gray, the E and the R, is because they're similar amino acids. They perform the same function, but slightly different. And just as a reminder, these parasites, reside differently in the body, but yet they have these conserved amino acids. And so what I wanted to test was what would happen if I mutated each of these amino acids separately and see the activity level. And that's what I did. Compared to the wild type, when I mutated the lysine to an alanine, we saw a dip in activity, but not abolishment. When I mutated the tryptophan to alanine, we saw an even lower, an even more decrease in activity, but again, not really abolishment. And then the other 2 amino acids, again, it decreased activity, but it seems as though with the other amino acids around, it's able to, compensate for the loss of this one amino acid. And so the next step was to mutate all 4 amino acids at once, take away this whole region, just these 4 amino acids, to alanine. And what do we see? A complete abolishment activity. And so we go from taking away the whole C-terminus to now just taking away 4 amino acids, and we get the same abolishment activity. I went one step further because this protein actually has a partner, a binding partner known as CREP-A2. And CREP-A2 is known to enhance function. And so what we see is with the wild type, as I add more and more of this binding partner, you get enhanced function. But when I add that binding partner, when I add that interacting partner to this KWKE mutant, we see a consistent abolishment activity, meaning that these 4 amino acids are not only crucial for the adenylation activity, but also it doesn't even enable its interacting partner to enhance activity or to help rescue activity. And so with that, I conclude that 4 amino acids, KWKE, are essential for adenylation activity, and thus an interesting drug target for Trypanosoma brucei and other parasites, as they have shown that are very conserved, and potentially an interesting region to target for cases such as that 67-year-old man that had human African trypanomyosis when visiting Africa. And with that, I'd like to pass it on to my colleague who would almost do something. He would go one step further. If this is the protein that we want as a potential drug target, how is it that we can potentially target it? How do we screen drugs to target this kind of protein and go forward in that research? So that will be talked about by some of my colleagues. But today, that is all for me. I'd like to thank you all for listening, and enjoy the rest of the talks. I'd also like to acknowledge all of my lab members, as well as my professor, Dr. Salavati, and the funding that I've received for this project. Thank you.